IB Bio, Option B, Biotechnology, Bioinformatics, Part 6, is the second of two movies that look at B.3, Biotechnology employed in the protection of the environment. Part 6, B.3B. This movie will have its sole focus on biofilms. The essential idea is biotechnology can be used in the prevention and mitigation of contamination from industrial, agricultural, and municipal wastes. Here is the outline of all the available movies for Option B. Use this outline to find the movie you need for review. This movie is focused here. Biofilms are an aggregate of bacteria and other microorganisms surrounded by a self-produced matrix that is attached to cells and a surface. Biofilms have formed on the teeth in this image. Teeth are one of the many surfaces on which biofilms will form. In aggregate, biofilms, like the one in this image, are more likely to cause harm, in this case cavities. Stay tuned. Here is a micrograph of Staphylococcus aureus in a biofilm. Staphylococcus aureus can be a virulent bacterium, often found in biofilms on surfaces in hospitals. In these photographs, we see a medical professional looking behind the eardrum in the ear of an individual. We see the normal appearance, and here, behind the eardrum, is a biofilm. Work to gain control of the definition of a biofilm, seen here. In Part 5, I spoke about Pseudomonas putida degrading methylmercury. The contaminated water is poured through a column on which the Pseudomonas putida forms biofilms. The biofilm of Pseudomonas, attached to the surface of the media within the column, serves to break apart the methylmercury, allowing the elemental mercury to be safely extracted. So we've defined biofilms. Now let's look at the formation of biofilms. The relevant IB syllabus statement is, describe how cooperative aggregates of microorganisms can form biofilms. Free-living planktonic bacteria adhere to a surface, reproduce to form colonies, and once the colony is large enough, and that's the key, the cells begin to produce a matrix within which the cells live, and it forms a film on a surface. In this diagram, you can see the free-living planktonic cells here. They attach to a surface and develop into a colony. Once large enough, and once there is a sufficient density of cells, the cells produce a matrix that holds the cells together and forms a film on a surface. Here is another diagram showing biofilm development, the planktonic phase to the adherence to a surface, to the development of a colony with a sufficient density of cells to produce an extracellular matrix within which the cells live and adhere to a surface. Here's one more diagram of biofilm development. I'll let you study this slide on your own. Now, biofilms are found everywhere, and that's what's interesting or scary depending on your perspective. Biofilms are found in rivers, lakes, soil, and within the human body, on contact lenses, lining the intestinal tract, as part of lung infections in patients with cystic fibrosis. And biofilms are found in the food production industry, on refrigerators, countertops, wine casks. And biofilms are found in hospitals, on surgical surfaces and surgical tools. Biofilms are found in the ventilation systems in airplanes. But biofilms can be harnessed to do good work, as I mentioned in the degradation of methylmercury by Pseudomonas, forming a biofilm within a separation column through which the contaminated water is poured. More on this later. As an aggregate of cells, as an aggregate of different species, biofilms can be more virulent or cause greater environmental harm. In this micrograph, you can see a biofilm corroding metal. Alone, the cells could not degrade the metal, but as a film of cells, a film of high enough density, they can. Here is a sewer pipe clogged with a biofilm. The biofilm is corroding the metal of the pipe. In time, the pipe will need to be replaced because of biofilm development. So I've been hinting at this IB syllabus statement when I've been speaking about the aggregate of cells in a biofilm, the aggregate of sufficient density, having greater virulence than the individual cells alone. Here's the statement. Biofilms possess emergent properties. Emergent properties are defined as a property which a collection or complex system has, but which the individual members do not have. In complex systems, new properties emerge that were not present at the simpler levels of organization. The new properties are emergent. Let me just strengthen the concept of emergent properties. The functionality of this mallet emerges from two component parts, the head and the handle, neither of which have functionality alone. The functionality of the flower to attract a pollinator emerges from the collection of petals, or emerges from the collection of cells that make up the petals. Vibrio fischeri is a species of bacteria found in aquatic systems. Vibrio fischeri often lives in association with other large living organisms. Vibrio fischeri is capable of bioluminescence. With high enough density of cells, bioluminescence appears as an emergent property. Emergent behaviors of biofilms include the production of extracellular substances, the matrix that I've mentioned earlier in the movie, resistance to antibiotics, increased virulence, and bioluminescence. When the density of cells in biofilms is high enough, as can be seen in this photograph, emergent properties appear. So the appearance of emergent properties, when the density of cells is sufficiently high, brings us to the next IB syllabus statement. Microorganisms and biofilms cooperate through quorum sensing. Quorum sensing is the regulation of gene expression in response to change in cell population density. In quorum sensing, the cells are capable of synthesizing and releasing signal molecules seen in this diagram as red dots. When the cells are in low density, there is no quorum sensing because without a high enough cell density, there is not a sufficient release of signal molecules. But as the density of cells increases, the autoinducer molecules, the signal molecules, increase in concentration, changing the gene expression within their own cells as well as neighboring cells, and emergent bioluminescence begins. This diagram displays a slight bioluminescence. When the cells are in high concentrations, the high concentration of the autoinducer molecules targets the genes in all cells, thus inducing the genes to express. This results in the full expression of bioluminescence. When the colonies have a large enough cell density, quorum sensing occurs and emergent properties appear. Quorum sensing is the regulation of gene expression in response to a change in cell population density. The emergence of new group behaviors occurs because of quorum sensing. Bioluminescence is an emergent property that occurs because of quorum sensing. Bioluminescence is not the only emergent property that results from quorum sensing. There are other emergent properties that come from quorum sensing that you need to bring under your control. So, for example, microorganisms growing in a biofilm are highly resistant to antimicrobial agents, in other words, antibiotics. I used the term virulence earlier in the movie. Microorganisms growing in a biofilm are more virulent. Biofilms on teeth, as you can see in this image, are composed of different species of bacteria which together, as a film, are more likely to cause cavities. When biofilms form on teeth, it becomes more difficult to remove them with antibacterial toothpastes or mouthwashes. Remember that quorum sensing involves signal molecules that serve to develop the colony, but also the signal molecules regulate the expression of genes that promote emergent properties such as resistance to antibiotics. Biofilms will develop on surfaces within hospitals, and as films, the colony will be more resistant to antibiotics. Antibiotic resistance is one of the top concerns of the medical community. This diagram shows a biofilm on a catheter, seen here, a catheter used to deliver intravenous medications. Biofilms, like this one, could result in infections unrelated to the reason a person went to the hospital in the first place, and the bacterial infection might be resistant to antibiotics. Biofilms resistant to antibiotics are likely to reside in our mouth, on catheters, and on the surfaces of artificial implants. Staphylococcus aureus is a pathogenic bacteria that will grow in biofilms. A micrograph of Staph aureus in a biofilm is seen here, and the films grow on medical implants as seen in this diagram. If antibiotic resistance is an emergent property of Staphylococcus aureus biofilms, then medical institutions need to be particularly vigilant in how hospital surfaces are cleaned. In addition to the emergent property of resistance to antibiotics, biofilms cause environmental problems. The IB syllabus statement is, describe environmental problems caused by biofilms. Drain pipes carrying sewage through urban centers can become clogged or corroded by biofilms. Eventually, the pipes will leak due to corrosion, resulting in urban health issues. In this micrograph, metal is being corroded by a biofilm. As you might imagine, biofilms form in industrial food production systems. Where there are organic molecules as a potential energy source, there will be microorganisms. In this image, we can see cheese being produced. Biofilms will form within the deep tank fermenters in cheese production systems. You should recall deep tank fermentation as part of Option B, Part 2. On this slide, we see biofilm formation in industrial kitchens, biofilm formation in the ice machine, and Bacillus cereus biofilm on a stainless steel surface. Didymosphenia geminata is an algal species that forms biofilms. Didymo is transferred among bodies of water by boats. Boats and shoes move didymo spores from one body of water to another. Didymo is a biofilm-forming invasive species. Invasive species outcompete native species, often resulting in the local extinction of certain species. The introduction of non-native species, such as didymo, is an environmental issue. The transfer of ballast water, as you can see in this photograph, can move the microorganisms that form biofilms. Water taken up in one location when the cargo hold is empty is released in another location when the vessel takes on cargo. The transfer of ballast water from one location to another can move the microorganisms that form biofilms. And remember, biofilms are found on hospital surfaces. Catheters and surgical instruments are examples of surfaces on which biofilms can be found. And remember that antibiotic resistance is an emergent property of certain biofilms. This is scary stuff. And that brings us to an IB syllabus statement that I introduced in Part 5. Describe the use of biofilms in trickle filter beds for sewage treatment. Polluted water, whether it be sewage or water contaminated with methylmercury or oil or benzene, can be trickled through a system that has bacteria in residence. The bacteria can form films, biofilms, in the soil or on the roots of plants or within the filter column. The biofilms of bacteria can be used to clean the polluted water. This is also called bioremediation. Using plants to treat polluted water is known as phytoremediation. As the polluted water moves through the bioreactor, the trickle bed with biofilms of bacteria, the bacteria metabolize the pollutants, rendering the water clean. Here is an industrial bioreactor. Polluted water particularly sewage, is pumped in at the top. The sewage water trickles through the bed composed of a medium of stones or plastics on which biofilms exist. Biofilms metabolize the pollutants to clean the water. Here's another image of a trickle bed bioreactor that uses biofilms. There's a fixed bed of rocks, gravel, ceramics, or plastics. The sewage or pollutants flow downward. Biofilms develop on the media. Aerobic conditions are maintained, and the water is cleaned. Here is an industrial trickle bed. You can see the sewage water being sprayed over the bed. Biofilms form on the medium of the bed and serve to cleanse the water. Remember Pseudomonas putida and its degradation of methylmercury. Engineers have designed filters with high surface area on which biofilms can grow for the purposes of cleaning sewage water. As well, bacteriophages are used in the disinfection of water systems. Bacteriophages are a group of viruses that infect specific bacteria, usually causing their disintegration or bacterial cell death. Sewage water often supports numerous pathogenic bacteria. Bacteriophages, viruses seen here, are infecting a bacterium, causing the death of the bacterium, rendering the water safe to drink. In this diagram, we can see the bacteriophage virus infecting a bacterium and causing its disintegration, degradation, cell death of the bacterium. This serves to clean polluted water of pathogenic bacteria, bacteria that cause disease in humans. Here is a diagram of bacteriophage viruses infecting and killing bacterial cells. Bacteriophages are used in the disinfection of water systems. And that brings us to the end of IB Bio, Option B, Biotechnology, Bioinformatics, Part 6.